I'm Nirmala. I work at Genome in Singapore and Prabhakar's the Computation Systems Biology Group. Today I want to discuss about one of our works in genome landscape and anti-depressant actions. Our project involves understanding what are cell types and what brain regions that play a major role in regulating any antidepressant action. For that, we are using a multimodal comprehensive approach. And I want to talk to you about more in detail about the cell work that we've done in conjunction with the work. Before I move on, needless to say for this audience, that depression is a major health burden. It's the second leading disability cause of disability by this year, according to the report. And depression is a whopping 350 million individuals. And quite radically changes the brain. You can see this PET that you see where the brain activity as function and structure is divided between rest and a rest brain. So therefore, it is very important to understand which are the brain regions affected, what are the underlying cell types that could have been changed. So several theories that the neurobiology of depression. One theory, monoamine hypothesis, refers to the major modes or the neurotransmitters we have. So healthy brain, two neurons, which is, you can see, there's serotonin molecules at the synapse, but in the case of a depression, there's reduced amounts of serotonin available. It gets recycled back by this presynaptic neuron. And this is one major hypothesis for why brain is affected by the depression. And antidepressants are drugs that will bring back the serotonin levels, higher serotonin levels at the synapse. So among the antidepressants, there are different classes of antidepressants, but the most widely prescribed and the dominant class of antidepressants are the selective serotonin reuptake inhibitors. So by the name, as the name suggests, so it helps to stop the serotonin from disappearing from the synaptic cleft. Okay. So SERTs or the SSRIs, there are many different kinds of SSRIs. They bind to this SERT transporter or the serotonin transporter and stops the serotonin available at the synapse from going back into the presynaptic neuron. So among all the SSRIs, the most widely prescribed SSRI is Prozac. Prozac is also known by its common name, its chemical name, fluoxetine. 250 million people worldwide are being prescribed SSRIs, and of that, Prozac is the most major leading SSRI that's been prescribed. But Prozac has certain downsides or side effects, adverse side effects. It affects, because these antidepressants target the many serotonin receptors, as a result of it, the side effects are like anxiety, nauseousness, sexual dysfunction, and ideas of suicide. It's quite ironic that an antidepressant would cause a suicide ideation, but these are the side effects of commonly used SSRIs. So therefore, because of the side effects and because of not a fuller picture of understanding the mechanism of action of SSRIs, it becomes imperative that we design better model studies to understand SSRI action. But as of now, majority of the SSRI studies are on rodent models for a good reason. They are good models to study antidepressant action as well as depression. But it would be good if we could go beyond the rodent models and complement the rodent studies with the primate models as well. And also another side effect, another downfall of the current trend is that existing studies only look at a gene-centric pathway, only one pathway or a few candidate genes, and it's focused on a few brain regions. Like, for example, the most majorly implicated regions are hippocampus and amygdala because they control mood. All right. So the downfalls right now are that we need better models. We need an unbiased systematic omics study, right? A more genome-wide study, and we need to look at many other brain regions. So that's exactly what we did in our study. So we started with a rodent model, which is, as I said, a good model to study antidepressant action. So we treated rats with fluoxetine and compared them to the control rats. Then we microdissected 27 brain regions, and we did a chromatin as well as a transcriptome landscape. We did bulk RNA-seq as well as single-cell RNA-seq. The bulk is limited. 27 brain regions were profiled by using the ChIP-seq and RNA-seq assays, whereas 2 brain regions were profiled for single-cell RNA-seq. And from that, the idea is we deduced 14 maximally informative brain regions, and then we took them on into the primate study. And then after validation and understanding the mechanism of action, once we've identified the genes, the loci, and the cell types, we come back to the 3 species, monkeys, rats, and human, and we integrate the data that we have, which is at 2 levels, one the epigenome and the transcriptome both at the bulk as well as at the single cell level. So the hope is that by integrating this data and finally we could infer shared and primate-specific molecular signatures of fluoxetine action. But why is this important for human cell atlas, right? So human cell atlas, this sort of an integrative analysis of multi-species as well as multimodal, even in the control rats, could add more value to the human cell atlas. And having an added advantage of understanding antidepressant action is a good addition to the existing resource or the data set that we have. So these are the 27 brain regions that we profiled. The brain region spans across right from the forebrain till the hindbrain. We cover most of the brain regions, widely studied regions, as well as regions that were not looked before. So what are the questions we want to address? We want to study the mechanism of action of SSRIs, yes, using 2 different model systems. But the questions we want to ask are, which is the brain regions that's most affected in the context of well-studied as well as not well-studied regions? And then the next question that we want to ask is, what is the genome-wide drug response? What are the molecular pathways that are involved? And what are the cell types that are involved in this response? And if we get lucky, the third question is beyond the scope of the study, but if we hope that we will be at least able to propose mechanistic hypothesis to understand what are the side effects of SSRIs. So a little more deeper into the study design. So we have rats from whom we give a brain, and then we do micropunches. And after micropunches, we have the RNA-seq, ChIP-seq, and single-cell RNA-seq. And from what do we expect to learn from this data set is that RNA-seq will tell us which brain region is important, what are the important DE genes in each brain region. ChIP-seq will tell us epigenome alterations, and single-cell RNA-seq will be a nice addition and complement the bulk studies by pointing out the key responder cell types to fluoxetine. So for single cell, so here onwards, I'm only going to take a deep dive into the single cell part of the study. I'm going to ignore the bulk ChIP-seq and the RNA-seq, that's for another day. But today, at this meeting, I think you would be more interested in the single cell. So what we did was we took 2 most important brain regions, the hippocampal regions, ventral dentate gyrus and the dorsal dentate gyrus, because they are known and they've been validated well enough to show that they are very major zones of neurogenesis. And antidepressants actually cause neurogenesis, and neurogenesis is seen to be the link for the brain to recover from depression. Right, so that's exactly why we chose these 2 brain regions. And here I just want to tell you that we had good quality cells. We discarded cells which didn't fit our cutoff, and we took good quality cells, and everything looks normal. The data QC looks normal. So with that assurance, we can move into looking into the data and gleaning insights from the data. So as all of you know, the first thing was just to annotate major and minor cell types. We used a reference-based annotation. So it was not as unsupervised clustering. So here we used RCA, reference component analysis, and you can see that the data set nicely clusters into 6 major clusters, 7 major clusters. There's one cluster that's unresolved, but we match it on to fact-sorted population to get these major cell types first, and then dig deeper, and then we use our unsupervised clustering to get the subtypes from these 7 major cell types. So this is just a new map showing the major cell types that we detected. The major cell types are the common, the usual suspects. And so we used RCA, a quick plugin. So Florian had probably given his talk earlier today during the lunch break. His talk is about the reference component analysis and how that could be used. The code is available on GitHub. And tomorrow, Bobby is giving a talk about DubStepR, the algorithm that helps you identify feature genes so that you can use those feature genes to perform your unsupervised, your clustering, your further subclustering. So do tune in for their talks. So we clustered 7 major cell types, and then with that confidence, we could subcluster each of the major cell types into further subtypes. And that's what we've done here. I'm not going to bore you with the details that of the subclusters, but we identified close to 15 to 17 subclusters. And so the first question is, do cell proportions change in response to fluoxetine? I'm not going to give you a more detailed microscopic examination of this, but just to point out, you could see that the sham is the control, fluoxetine is the disease-treated, drug-treated group. You can see that this gray population decreases in one brain region, whereas it increases in the other brain region. So this nicely captures the region-specific effect of the drug, right? And also there's another subtype with a similar trend, where it expands upon treatment with fluoxetine. So yes, there are some cell proportions that are changing. So we are validating this finding in a different independent cohorts before we can safely conclude that. So the next question that we asked was, what is the key responder cell type to fluoxetine? So there are many ways to answer this question, but one way is just to look at the differential genes. Which cell type has the largest differential genes, either the largest number or the largest effect sizes? And as you can see here, the differential genes from a dorsal dentate gyrus, the dorsal hippocampus are marked in blue. The genes that are the differential genes from the ventral hippocampus are marked in orange. So it looks like a lot more cell types from the ventral hippocampus are drastically changing compared to the dorsal, but the largest effect, the largest changes are nonetheless seen in the dorsal dentate gyrus. Right, so with that answer, the next question that we asked was, well, we know that there are human GWAS for depression, for antidepressant response. So how does this data set, how does these individual cell types that we've identified map onto those GWAS hits? Like, are any cell types that we have, are the differential genes that we identified in any of these 15 to 17 cell types, are they specifically enriched for a particular GWAS? So as you can see in the dorsal dentate gyrus, it's the glia, the Bergman glia cells, as well as the neuron, a particular excitatory neuronal neurons are enriched for SSRI response as well as for depression, right? And in the ventral dentate gyrus, it's complete heterogeneous response, except for the Bergman glia. The enrichment of GWAS hits across cell types is different. And this is something that we are looking deeper into. And so what other things can we learn from the single cell study? So we had an insight from our bulk analysis, which was, we looked at the serotonin receptors and also the other neurotransmitter receptors. And what I've highlighted here is only HTR1A, which is the serotonin receptor 1, right? So it is downregulated from our bulk studies. So then we said, if our bulk study in this particular brain region, in the dorsal hippocampus, if serotonin receptor is downregulated, what is the status across the cell types? Is there a heterogeneity across the cell types for this particular receptor expression, or is it going to be uniform? That's the next question we asked, and I'm only giving you a snapshot here. So if you look into HDR, if I can draw your attention to HDR1A, there is heterogeneity in the response between the different cell types. So the x-axis are the different cell types. So you can see that certain cell types are upregulated, certain cell types are downregulated in conjunction with the bulk. But just that the bulk data set averages out all these different subtypes, and so therefore what you read is a downregulation. But our single cell data set helps us identify this heterogeneity between the different cell types. Another insight that we learned from our bulk analysis, so what I'm showing you is a heat map of the k-means clustering of the ChIP-seq peaks, the H3K histone, H3K acetylation peaks that we did after treating the rats with fluoxetine. So we looked at what are the chromatin changes that this drug causes, right? And we identified a very peculiar cluster, specific only to the dorsal dentate gyrus. And so we dug deeper into it, and we looked at what is the motif that is enriched in this particular cluster. And we found out that NeuroD1 is the highly enriched motif with a significant p-value, with a more significant p-value. And so therefore, what we did is then we came back into the tissue sections, and then we did a single molecule RNA fish at the control and the drug treated sections. And what you see here is that you see that there is an increase in the expression of NeuroD1 RNA levels and also BDNF. So BDNF is a direct target of NeuroD1. So NeuroD1 is a transcription factor that causes neurogenesis in the hippocampus. So this is a positive control, but also another added layer of new validation that this data set brings in, that yes, neurogenesis happens in response to fluoxetine, and it happens by altering the NeuroD1 levels, which is a transcription factor required for neurogenesis. So because we have a single cell data set, the next question that we asked was, is this NeuroD1 heterogeneous, or can our single cell data set identify which cell types are preferentially upregulated for these neurogenesis markers? And that's what you see. So what you see here is just a ridge plot, a distribution plot of the expression for the NeuroD1 targets. Orange denotes sham, and fluoxetine is by blue. And so true enough, I've only listed two subtypes here. We identified two subtypes that we identified from our single cell RNA-seq that show a differential response in NeuroD regulation, or the entire set of NeuroD as well as its downstream target genes. So we are validating this currently to see if we can get a mechanistic insight and also identify any novel gene candidates and drug targets here. So I want to quickly summarize that what we have out of this study is a genome-wide landscape of fluoxetine-induced changes. What I gave you is just a primer. The manuscript is in prep and will soon be up on bioRxives. So what we have is a large data set of ChIP-seq, RNA-seq, and single-cell RNA-seq. In the single-cell RNA-seq, we have about 500 genes that are differentially altered across cell types. And we've identified key cell types, and we are validating the molecular pathways underlying the fluoxetine action. As I already said, deeper dives are ongoing, and the future direction is for us to bring in the human data set, the postmortem brains from depressed patients, from healthy patients, as well as from patients that were depressed patients that were treated with SSRIs, or more specifically fluoxetine, and do the integration to see how much we can learn across the different species and the different multimodal analysis that we have done. And with that, I want to thank Shyam for having provided me this opportunity to work in his lab and for his guidance, and Michael Meany for his profound insight and direction in guiding this project, and a whole bunch of extremely talented and highly vivacious group members and co-authors who have helped me get this project off the ground. And besides that, every member of the CSP6 lab. With that, thank you. And I'll take any questions.
Okay, we still have a couple of minutes for question, and also you can pull out the question from the chat room.
Yes, as we are speaking, I'm doing that.
Okay, great.
So there are two questions, I think, on this. If you pull out the note, this when did you analyze mouse brain after SSR1 treatment? How many days after?
Yes, yeah, great question. So we did a long-term effect. We looked at the long-term effect of fluoxetine. So it was 8 weeks of treatment.
Okay, so you already see the SSR1's function effect after 8 weeks, right? Only after you see the effect, you start to do the.
No, we've compared. We've done a time comparison as well. So we've done SSRI. We've looked at SSRI function after 3 weeks and then after 8 weeks. But the data that I presented for the single cell is for a long-term treatment, which is 8 weeks.
Okay, I think the attender also can raise hand to ask questions directly.
Oh, I see another question. How many days are required to see the effects of SSRI? Yeah, so that is one of the major disadvantages of SSRI. SSRI's action is clinical effect is not seen within days. It takes a couple of weeks. So it at least takes 2 weeks for us to clinically see the improvement in humans. Is it easy to collect depressive human brains? It is not easy. It's a very great question. It's not easy. That's exactly why we leverage on our study on rodent models as well as primate models, because it's easier for us to get the material required for the work. But we do have access to a human brain bank, a depression human brain bank, through our collaborator, Michael Meany. Yes, so we have limited brains. Yeah, we do have access.
So okay, if other people have a question, just submit and we can. I think we can just answer by email or by chat room something. I think the time is up. We would like to thank all the 3 speakers, including me. I'm just like, that's kidding. And I think it's a great time and as a great opportunity to share what we did. And okay, yeah, let's close this section. Okay, bye.
Right. Thank you.
Bye.